Immunology Insights, Basics and Beyond. Chapter 53. Luc Montagnier and the Discovery of HIV. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored how the immune system responds to major viral threats like influenza, Ebola, and SARS-CoV-2 pathogens that reshaped public health and taught us how fragile and resilient our immune defenses can be. Today, we revisit one of the most extraordinary detective stories in medical history, the discovery of the virus that causes AIDS, the Acquired Immunodeficiency Syndrome. It's a story that began in mystery, unfolded through scientific rivalry, and ended with breakthroughs that changed the world. At its center stands Luc Montagnier, the French virologist who uncovered HIV and forever altered our understanding of the immune system. So let's talk science. Let's have first a look at the early days of a mysterious epidemic. In the early 1980s, physicians in the US and Europe noticed a terrifying pattern. Young, otherwise healthy men, often in urban centers like New York, San Francisco, and Paris, were dying of infections and cancers that typically only affected the severely immunocompromised. Plymocystis pneumonia, Kaposi sarcoma, and rare fungal and viral infections appeared seemingly out of nowhere. Initially, no one knew what was happening. Some speculated it was an immune toxin, others blamed lifestyle factors or even immune exhaustion from chronic infections. There was confusion, fear, and stigma. Public authorities were slow to react, and the disease became tragically politicized before it was even scientifically understood. This uncertainty drove researchers worldwide to search for a cause. Among them was Luc Montagnier at the Pasteur Institute in Paris, a retrovirologist known for his precision and patience, both essential traits when working with fragile viruses. Now to the discovery of HIV. In 1983, Montagnier and his team, including François Bar-Sinoussi, isolated a new retrovirus from the lymph nodes of a patient showing early signs of AIDS. They named it LAV, lymphoadenopathogen-associated virus. Using electron microscopy and reverse transcriptase assays, they demonstrated that the virus was not a known human retrovirus, but something entirely new. The virus targeted CD4-positive T helper cells, the commanders of the adaptive immune response, using the CD4 receptor and the co-receptors CCR5 and CXCR4 to gain entry. Once inside, it used reverse transcriptase to copy its RNA into DNA and integrate into the host genome, a hallmark of retroviruses. Over time, as these T cells were destroyed, the immune system's ability to coordinate defense collapsed, leaving the body vulnerable to even harmless microbes. Almost simultaneously, Robert Gallo's group at the NIH identified a similar virus, which they called HTLV3, so Human T Lymphotropic Virus 3. A high-profile controversy over credit followed, LAV versus HTLV3, but later sequencing proved both were the same, Human Immunodeficiency Virus, HIV. The discovery transformed panic into understanding and laid the groundwork for diagnostic testing, safe blood transfusions, and eventually life-saving therapies. Now it's time understanding the virus and its devastation. HIV's stealth lies in its persistence. Initially, it was not even clear how the virus spread. Today we know it is through bodily fluids, via unprotected sex, and unchecked transfusions. After initial infection, it spreads through lymphoid tissues, establishing latent reservoirs that evade both immune clearance and drugs. The body mounts a massive immune response, antibodies, cytotoxic T cells, but never clears the virus. The ongoing battle gradually exhausts the immune system. When CD4 positive counts fall below a critical threshold, the patient enters the AIDS stage, defined by severe opportunistic infections or certain cancers. These include Pneumocystis jirovecii pneumonia, Mycobacterium avium, Candida infections, Toxoplasma encephalitis, and Kaposi sarcoma. What kills isn't the virus directly, it's the collapse of the immune coordination. By the late 1980s and early 1990s, AIDS had become a global crisis, hitting hardest in sub-Saharan Africa and among marginalized groups in the West. Stigma, fear, and misinformation compounded the tragedy, but also ignited activism and an unprecedented mobilization of science. And then finally, the treatment revolution came. From fatal to chronic, the first antiretroviral azidotumidine, or in short AZT, emerged in 1987. But it was only partially effective and also toxic. The true turning point came with HART, the highly active antiretroviral therapy, in 1996, a drug combination that simultaneously blocks reverse transcriptase, protease, and integrase enzymes. These therapies shut down replication at multiple steps, keeping viral load undetectable and allowing immune recovery. Today, a single pill regimen can suppress the virus for decades, turning HIV into a manageable chronic disease. Preventive strategies like PrEP, so pre-exposure prophylaxis, and PEP, post-exposure prophylaxis, protect those at risk, while U equals U, so undetectable means untransmissible, campaigns have transformed public understanding and reduced the stigma. Mechanistically, these drugs work by interfering with viral enzymes or blocking entry pathways, either preventing the virus from copying itself or integrating into the host's DNA. The next frontier is cure research, seeking to purge or permanently silence latent viral reservoirs in resting CD4 positive memory T cells, using gene editing, immunotherapy, or latency reversing agents. Such interesting stories for sure have also controversies, especially in Montagnier's later years. Luc Montagnier early career was marked by rigor and brilliance, but his later years took a more eccentric turn. He explored unconventional hypotheses, claiming that DNA could emit electromagnetic signals, that water might retain a memory of molecules once dissolved in it, and that low-frequency electromagnetic waves could influence biology. He even suggested that oxidative stress and bacterial imbalances could contribute to autism and chronic disease. These ideas, often unsupported by data, were dismissed by mainstream science, yet they reflected a restless intellect unafraid of exploring the unknown. Though many of these later claims fell outside scientific consensus, they do not overshadow his genuine scientific achievements or the millions of lives saved by his early work. Let's also look at Robert Gallo, who shares the recognition. Robert Gallo's role was equally critical. His laboratory confirmed HIV's link to AIDS, developed the first blood test to detect infection, and characterized how the virus replicates and spreads. Though the Nobel Prize in 2008 went to Montagnier and Bar-Sinoussi, Gallo's contributions helped shape the field of retrovirology and the global AIDS response. Both scientists, despite rivalry and political drama, ultimately thought the same goal: to uncover the truth behind one of humanity's greatest and medical mysteries. Of course, all this has a broader legacy and relevance. The discovery of HIV reshaped medicine. It catalyzed advances in molecular biology, epidemiology, and immunology, driving progress in everything from viral diagnostics to vaccine platforms. It also forced the world to confront stigma, inequality, and the human dimensions of science. The collaborative network built during the AIDS crisis later accelerated research during COVID-19, proof that lessons from one pandemic can safeguard us in the next.
To conclude, Luc Montagnier's discovery of HIV was more than a scientific triumph. It was a turning point for global health. From the fear and confusion of a mysterious new disease to the creation of life-saving therapies, the story of HIV and AIDS is one of science, courage, and relentless human effort. Montagnier's legacy reminds us that curiosity and compassion can overcome even the darkest biological challenges. In our next chapters, we'll take a closer look at allergy and asthma, beginning with the mechanisms of allergic reactions. This section follows our discussion of infectious diseases to show the other side of immune imbalance, how an otherwise protective immune system can overreact to harmless stimuli. In other words, we are shifting the focus from underreaction or immune evasion by pathogens to overreaction against innocuous antigens. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.